welcome back, everyone. Next up is Bioxytran Inc. They're an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. And they have a second platform drug, an oxygenation molecule being developed to treat stroke, and its development is supported by an FDA-approved device that detects local tissue oxygenation. So due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. And the company has a third platform technology that's capable of treating cancer metastasis. Now, Mike, nice to see you again. Before we get started with your presentation, I just want to let you know I've been following you and noticed you recently published another LinkedIn article on how to slow the aging process. Hello. Using the universal oxygen carrier. So what I found interesting is you mentioned the universal oxygen carrier was equivalent to 18 hours of hyperbaric oxygen treatment. So I know that's very popular right now at a lot of hospitals or med spas and smaller doctors. So do you think your oxygen carrier could eventually replace the hyperbaric chambers?
Well, Anna, this is a $3 billion a year business. And getting hyperbaric oxygen, it's really time intensive because for it to work best, you got to do it daily for at least an hour. That's a lot of time commitment when you're looking at these HBOT treatments, whereas the alternative is our universal oxygen carrier, and that's just like a shot. You get it, and it's going to give you the equivalent of about 18 hours of HBOT therapy. So if we get this universal oxygen carrier approved, this is going to be like a no-brainer for people because time is money. And you can spend a lot more time doing fun things versus just kind of sitting there in a chamber, right? It's for these reasons that I can foresee that almost all the hyperbaric oxygen patients are definitely going to much rather prefer the injection over the chamber.
Absolutely, they will. Well, thank you for clearing that up. And it also looks like you revamped your investor presentation. So when you are ready, the floor is yours.
Okay, well, thank you. Well, let's start off with our title, Taking Inflammation Out of People's Lives. So we want to kind of rebrand ourselves as an inflammation company. The next slide here is our forward-looking statements. And now let's look at the investing thesis for Bioxytran. Bioxytran has game-changing platform technology. We got 3 of them that we're going to go over today. We have clinical trial results that were, look, they're the best that are out there in the past decade. We got 100% responders rate. We'll go through all that as well. We have good IP, proprietary science, and bottom line, we want to make the world a better space. Now, this one bothers me. Our valuation is just ridiculous, and it shouldn't be here. But we're trading at an $8 million valuation, and I've been to a couple conferences and tell people about what stage you're at these conferences. These are professional investors. I don't think anyone's less than 200 million. And here we sit trading at $8 million market cap. So I think retail needs to step up, and I think we're at an ideal buying level. We also have a solid management team, and we have a solid strategy. We have a thin float, shareholder base. So, there's a lot of potential upside here. I just need people to realize that the final thing is we're in a lot of active licensing discussions. And that's why I've been going to all these conferences. We're a carbohydrate company. We make complex carbohydrates. What do? We're going to treat viral infections. We're going to treat cancer. And we have an oxygen transport molecule, and we stabilize it with our copolymer. And these carbohydrate drugs recognize galectin receptors, and they'll also carry the oxygen atoms. So now that I mentioned the word galectins, what are these things? Well, this is the definition, nice and big for you to all see. Galectins are protein receptors that recognize sugar molecules. So they're specially designed proteins that recognize sugar molecules. And here's the punchline. And they're responsible for chronic diseases. So it's the key to chronic diseases. That's why galectins are important. And then here I explain it a little bit more in depth with a little graphic there. And you'll see galectins, they're these structural things. You'll see the first one, they're kind of two Pac-Man behind each other, and the other one's a rod, and then the other one looks like a snowflake. And galectins stick to other galectins, and galectins also stick to those rods that are sticking up out of the cell surface. Those are called glycans. And you'll see like the middle one, it looks almost like an arresting wire. But they are also responsible for signaling. And galectins are extracellular and intracellular, but we're focused on the extracellular. Now, how big is the reach of these galectins? There's this article that says galectin-3, one receptor for an alphabet of diseases. Just take a look at all these diseases. I mean, for every single letter of the alphabet, there's a disease. I just want you to see that. We're going to just gloss over it kind of quickly, but there's a lot of chronic diseases here. So that takes me to my investment highlights here. So we have three platform technologies. The first one's a broad spectrum antiviral. Next one is cancer metastasis, as well as fibrosis. And then we have a blood substitute. And we are going to start, for those that haven't heard of us, we have an active FDA IND with our broad spectrum, and we finished a phase 2 study for antiviral. And for the blood substitute, we are in preclinical trials and quickly moving toward getting an IND. Let's take a look at prolactin M. That's our first, most advanced molecule, the antiviral asset. When I do this, what I'd like to do is, we have a historic discovery in virology. You see, for the past 50 years, there's this idea that one drug, one antiviral, and with our drug prolactin M, we've basically debunked that theory. We have what we believe is the world's first broad spectrum antiviral, and the discovery was outlined in an article back in August of 2024, and it basically talks about viral adhesion has to happen first. The virus has to adhere to the cellular membrane first. It's the first step in the viral entry process. So it comes in, you see, because what happens is you have these spike proteins, they're slippery. And why are they slippery? They're covered in sugar. And the reason they're covered in sugar is to be invisible to the immune system. Otherwise, the immune system would see them, right? And then you've seen cells under the microscope, they're slippery. So how do two slippery things stick to each other? They don't. They need help. And the viruses basically hijack these galectins in their environment and use them as adhesion molecules. And let's finish this slide up. So there's no broad spectrum antivirals that currently exist right now. And we're literally disrupting a $55.36 billion global antiviral market. So when I say our market valuation doesn't make sense, you just got to do the math. Here's an overview of our technology. And when we say broad spectrum, these are different families of viruses right there in the center. And you'll see that there's 3 that are circled. And no other antiviral has this sort of activity. And that's really what we want to point out. So we've done these 3 different families, and we plan on doing more to prove out this platform approach. And I'd also mentioned that I went over it fast, but there's 60. We have a patent for 60 viruses, or patent pending rather, for 60 viruses. We have clinical trial results, so let's go over them. We eliminated the virus and load within 5 days. We reduced infectivity, that's spreading it to one another. And we quieted the cytokine storm, and we have a robust antibody response. So we're gonna breeze through this. This chart on the right is basically just one line of the graph on the left. And what you really wanna see is there's that red line there, and all the green lines are the people that took the drug, right? And you want to be above that line as quick as possible. And you'll see that right at the 3-day point, right where it breaks, that is where almost everyone in the green was above the line except for a couple. So 88% were PCR negative. And then at day 7, 100% were PCR negative. But if you look here at the chart on the right, you'll see that 30% of the people were PCR negative, probably based on this curve, in about a day and a half. Do we have viral rebounds? No. Paxlovid does. We do not. And there's the chart. How about symptoms? Do we improve symptoms? Yes. And you'll see what happened is the top part of the chart is basically the baseline, and the bottom is at day 7. So when you look at the baseline, there's an even distribution between the blue, which is the group that took our drug, and the orange. And then you'll see nothing but orange. I mean, just a teeny weeny bit of blue there, but pretty much nothing but orange, and that denotes symptoms. So it followed, not only do we get reduce the virus, but the symptoms went away. What about a response? What about a long-lasting response? That's what this is, this idea of post-infection immunization. We built a robust response, and this is what this graphic shows you. Now, I'm dying to get to this one because this one shows how we are. This is a comparison, but it's not a head-to-head comparison, and because actually unfair to us. We didn't do a trial with Paxlovid. We took their real-world data and compared it to our clinical data. And what you'll see is there's this line of demarcation at the 30% level. How many people can get to the 30% PCR negative level? And if you look, it takes us less than 2 days to cross that line versus Paxlovid, which is 20 days. So I love to say, do you want to be sick 2 days or 20 days? It's almost a no-brainer. Well, how does this drug work? Well, we neutralize the spike protein. See, typically what we're showing here is the antibody comes in. You see where the purple meets the green on the top? It comes in there, binds to there, and that prevents the virus from entering. That's the way you typically do it. Well, our molecule, it basically goes into that galectin fold. It's circled there in red on the left, and it goes in there and it binds to that, and it prevents the spike protein from entering. And then what you'll see is the sugar surrounds the spike protein. The spike protein on the left in light blue, it's naked, and then the way it exists in nature is with sugar on it. And the sugar hides it from the immune system. And you'll see the tip is never covered in sugar, because if it was and it was hiding, it wouldn't be able to enter. And then, of course, the galectin fold. So we neutralize the antibodies, or we neutralize just like antibodies neutralize. And how does it get rid of things? Well, what we do is we gather, our molecule gathers up the virion, and it latches onto them by the spike proteins, and then travels through the body, eventually reaches the liver, and the liver purges it. Now we have other indications. Shingles was one indications there. You're going to see the before and after picture. This was a topical cream or paste that you put on, and you'll see how quickly it reduced it, and it got rid of the pain. And then conjunctivitis was another case study that we did. So there's other indications. We could use this for long COVID. We have a special long COVID deck, so we're just hitting the high points. The other thing, the next slide talks about bird flu. There's a very good case that this could work on bird flu, and we're actually pursuing some strategic relationships right now. Working on signed NDA type stuff in bird flu. This is something. This is Ebola. This came out of a BARDA slide deck. I just want to show you some of the things that we're working on. So a couple of decks, slides from our presentation decks made it in here, and we'll talk about cancer in later on. And the bottom line is this is an enormous market. There's no effective treatments out there for these viruses, and we believe we can make a difference. So now let's get to the next platform technology. We're talking about neutralizing cancer metastasis and fibrosis with prolactin I. Here's an overview of our platform technology. So we have an IV drug that can target cancer metastasis and fibrosis. It's designed to go after metastasis in late patients. And we also have this other thing, and that's the second bullet point, is combination therapy to enhance existing checkpoint inhibitors. We're going to really talk about that and why that's important to us as a company. And then combination therapy. Galectins could be used in combination therapy with almost any treatment options out there. And that is also in a cancer deck, specialized deck on cancer. So go to our website and take a look at it. Galectin-3 is a target, strong potential in cancer since 2018. And let's look at some studies and let's build a case. High galectin expression leads to bad outcomes. And this chart takes a little bit of explaining, but the bottom line is that people with high galectin expression, that's bad. If you're a person who has high galectin tumor staining on your tumor, that's not good. And you'll see what happens is the people fail. I'm summarizing what's going on in this slide. The people fail, the people in red, they fail really fast, like 30, 60 days. They can't take Keytruda treatment. But look, 100% of the people that had low galectin expression had good outcomes. So the question I pose is, what if galectins were blocked? Theoretically, Keytruda could be 90% effective in non-small lung cancer if you exclude people for high galectin-3, according to this peer-reviewed study. So let's dive a little bit deeper. Why is that? Well, a couple things. Galectin effect, T-cell anergy. Again, all this is covered in my cancer deck, so I'm going to kind of go over it fast. There's T-cell anergy. Basically, the T-cell goes into this wave of, we'll say, galectin, this galectin cloud before it gets to the T-cell, and it just gets all this plaque on it, on the actual T-cell itself. And what that does is it prevents these little grappling hooks, which are called lectins, and they grab on and get good adhesion with the cell, so they're mechanical, and they're basically plastered in concrete. And we go over that in the cancer deck. And the other thing is T cell receptor downregulation. Well, if you don't have as many probes out there on the T cell looking for these cells, you're not going to find as many. And that also happens thanks to galectins. What else does galectin do? Well, galectins are also reprogram the tumor vasculature. What the heck does that mean? That has to do with the blood flow. So you have a tumor here, and you have a blood supply going to it, and it grows. That's called angiogenesis. It grows inside the tumor and feeds it with oxygen and nutrients. So galectins are responsible for that, and they're responsible for every part of the angiogenesis process. So when you block lectins, you stop that, or you inhibit that. And that's another key point. And then this also tells you this mechanism, again, go to the cancer deck, but I'm going to sum this up. It basically gets you into this non-inflammatory environment, this suppressive environment where it suppresses your immune response. And that's what this chart talks about. And then what we did is we did an article, and this is the abstract of our paper. We did an article on immune checkpoint resistance. You see, why is this so important is because it is the part on the right. Overexpression of galectins drives several key cancer progression mechanisms. And you can see almost all the things to do with cancer involve galectins. So if you block galectins, we're expecting good outcomes to happen. Galectins are essential for survival of the tumor. So this is an article, it's a journal article. Again, let's not spend time on it, but we figured out why all these immune checkpoint inhibitors don't work as well as they should. I mean, they cure some people, but then why don't they cure the other people? This discusses this. And what the interesting one is in the upper left quadrant of those four charts there, it shows there's a blue line. So when you go to the cancer deck, look at it, there's a blue line there. And it basically, galectins completely render Keytruda, neutralize it. Basically, that's what it is, neutralization of Keytruda. So Keytruda is being given to a person, and it's just not going to work, according to this article. And what happens if you block galectins? Here's the schematic: is you rip these galectins off so Keytruda can now bind in its binding site. You see what happens? These galectins were occupying the binding site, and now Keytruda comes in. And that could end the resistance. And here's another article that talks about it. And when you add a galectin antagonist, you see over here on the left, when you have a suppressed immune system, and then you get Keytruda, and then it activates it a little bit. But when you get Keytruda plus the galectin antagonist, it's completely activated. That's what this chart is basically showing. But now this is a great slide. Why? Because it shows you this is in pancreatic cancer. It's a Kaplan-Meier survival chart. But basically, you want that chart to be pegged right to the top, like you want it to be at 100%. Well, it's not at 100%. There's a little bit, because that would represent 100% survival rate of all the people. And then the way this works is there's 5% survival if you have a pancreatic cancer versus, I think it's 65 or 68% if you're galectin-1 negative. So if you don't have galectins, that's good. There's 68% chance you're going to survive rather. If you do have galectins and you're positive for them, it's about a 5% chance you're going to survive. So the whole idea is just makes sense to block lectins. And then here's our clinical evidence. There's 2 clinical trials, reduction in immune side effects. All this stuff is on my cancer deck, but it's just worth looking at this picture here. This is a before picture. The guy on the left, baseline, and then completion of therapies. That was 90 days. And of treatment, 90 days of treatment, that's it. And then they stop. And the guy got a complete response. So when you compare that person, and he failed Keytruda a couple times before, and all the tumors were resolved. This is incredible data. And you know how old this data is? This data dates back to 2018. We think it's time that galectins are going to make it to the forefront. And here's another galectin antagonist. And both of these are not ours. So I want to be clear about that. They're not ours. But we have a common target, which is galectin-3. And look, at the lowest dose, 40% achieved a partial response. And then they upped the dose a little bit, and then it went to 60. Well, that's combination therapy. Well, how does that compare to the standard of care? It's 22 to 38%. Well, let's just call it, call it 30%. Look at the numbers. So even at the lowest dose, you're better in combination, and you're twice as good in combination at the higher dose, and they didn't even do the next dose escalation. So we don't even know how that would have worked. And then this is the checkpoint inhibitor landscape. And now I want investors to just pay attention real hard here. This is our target market. These are the 11 guys that I'm going to be talking to. And I got a couple of them that I'm already in. I have phone numbers and stuff. And I'm making my slide deck and my presentation is to talk to these guys. This is who we want to do a deal with, because these guys need us. They need what we have to remain competitive. All right. And I put it in blue. The pharmas with galectin-3 blockers are going to dominate oncology. We are sitting in a power position, and we have an $8 million market cap. Prolactin I, that's for fibrosis. I just want to bring it. I want to bring it in. The same molecule will work on cancer and fibrosis. Again, it's this inflammatory cycle that's driven by galectins. And this inflammatory cycle, you're gonna, when you go to the slide decks, take a look at this. What you see is this galectin. It's at the center there, and it's the center of these feedback loops, these pro-inflammatory feedback loops. And you even have TNF alpha over here. Some people think TNF alpha is the big thing in inflammation. It's not. It's actually galectins. Galectins have a bigger impact in these diseases. And we're running out of time here, so I'm going to go through my blood substitute as fast as I can. We also call it the universal oxygen carrier. It's our third platform technology. Look, we have proof of concept in animals. We have 2 of the 4 animal species tested. There's no change in side effects. Here's an interesting thing. Government discussions are being pursued. We got one tomorrow, as a matter of fact. Let's show you how it works. The universal oxygen carrier works as a bridge. It's 1,000 times smaller than a blood cell. So you'll see this blockage here in this picture. So the blue's going through, no problem. Obviously, no red blood cells are going through. Well, the blue is carrying oxygen, and that's the idea behind it. And then the way we make it is put in a little graphic there. You see what we do is we extract these heme molecules, and then we stabilize it with our copolymer, and that's how we make it. But we have some incredible data here, and we presented this to BARDA, and this is some data. It's old data. It doesn't use our molecule, but uses the same chemistry. We used a hemorrhagic model. We took out 90% of the blood out of these dogs. And what happened is you see something you just don't expect to see. The tissue oxygen tension, the TPO2, it actually went up. And the model is supposed to have a 50% mortality in 2 to 3 hours. And at these high settings, or at the high dosage levels, and that's in the center one, you'll see that it was statistically significant. So we actually had a survival benefit in this study. Just miraculous stuff. And this shows you what happens with mice. These mice, what they did is they clamped off an artery that was going to their vein. And what happens is you see the white area there, and then that's what happens normally. But now we add our molecule to it, and it's a molecule with similar chemistry. And what happens is it goes right through the clot, and you see the pink tissue on the hemispheres. See that? And
All right, I'm sorry.
I think we're going to wrap, huh?
Yeah, it's time. I mean, good ending with that mouse slide, but it is time to wrap it up. Really fascinating as always. We've got so many questions for you, but we'll have to send them to you so you can answer on your own time. But thank you for this great update.
Wonderful.
All right, well, we'll see you again soon. And everyone stay with us. We'll be right back.